All right, Pirates, welcome to our second session of notes. Make these notes a little bit bigger, a little bit easier for you to see, and I'll simplify them a little bit more so you don't have as much writing to do. So today's notes are about DNA replication. Okay, so you might be asking yourself then, let me focus on this a little bit. There we go, nice and easy. All right, so what is DNA replication? What is it? Why do we have DNA replication? Well, the word replicate means to copy. So all this is simply making an identical copy. So simply making an identical copy of DNA. And why does it have to make an identical copy? Well, it's so it can pass on a copy to a new cell. So this occurs during cell division, mitosis, meiosis, etc. All right, so replicate means to copy. There's replication, it means copying. So simply you're making an identical copy of DNA. When you replicate something, you copy it. So where does this occur? Okay, where this occurs is the nucleus. Okay, the nucleus is the control center of the cell. It is where the DNA is located. Okay, so that's where the DNA is located. Anytime you have DNA involved in a chemical reaction, it's going to take place in the nucleus. The nucleus protects the DNA from mutations and protects the DNA from being scrambled so that it can go ahead and do the job that it needs to do, which is to create all the proteins, and then the proteins go on to create the organism, which is you. All right, so the nucleus you can kind of see under a microscope sometimes depending on what type of cell it is and how powerful the microscope. It's usually toward the middle of the cell. Almost all of your cells have a nucleus. And you can actually sometimes see the DNA. It kind of looks like a little cloudy area in the center of the cell. So you have a lipid bilayer, a little pouch, a little bubble, all the organelles, all the cytoplasm, all of the proteins are in this part of the cell outside of the nucleus, and then inside the nucleus, that's the main thing that's there, is the DNA, and the DNA just hangs out right there in the nucleus. So whenever you have DNA involved in a chemical reaction, it's taking place in the nucleus. That's the only place DNA can be found. All right, so you'll need to know this part here. It happens in 3 steps, and I don't know why I put a hyphen there. I'm going to delete that hyphen a little bit there. The first step is it unzips or unwinds and unzips. So as we saw in the previous, actually there's only one P in unzips, my bad. All right, so it unwinds and unzips. As we saw in the previous notes, DNA is a double helix. It's all twisted around upon itself. And so that DNA there, that is protected. It's small, so it can fit inside the cell. We have the DNA like that. So the first thing that happens is it unwinds, and it unwinds back into that ladder shape that we talked about. Remember, you have your phosphates and your sugars along the backbones. The rungs of the ladder are the nitrogen bases. All right, so I'm going to zoom in on those strands there. Sorry, my kid in the background playing. I'll make up some bases here. T, A, G, G, A. Tagga. All right, and then the complementary base pair rules. T always goes with A always goes with T, G always goes with C, and A always goes with T. All right, you're going to have different codes. Remember, this strand is the coding strand. This strand is the complementary strand. And remember, these two strands are held together using a dotted line here, unlike the solid line that I used for the covalent bonds in the backbone, because that's the hydrogen bonds. And that's where the DNA unzips. Okay, so the DNA unzips, the two strands separate, and you get your 2 individual strands. I'm going to move them apart a little bit, but it's still the same code on each strand. So this strand going down is still T, A, G, G, A, and this strand right here is A, T, C, C, T. Notice these codes don't match up. They're not the same code. They complement each other once again. So now we have what was one ladder here. The two strands are now separated, and that's important because right here is where the next step is going to occur. Okay. Step number 2, and I'll put these notes on Schoology in PDF form for you guys to look at. The second step is to add complementary bases. Actually, we'll say nucleotides because that's going to be more correct. Nucleotides, because what you're doing is you're adding new nucleotides that complement the inside. The bases complement each other. You still have the phosphate and the sugar. All right, so you add complementary nucleosides along the inside. So what we did is I'm going to redraw my two strands that I had previously. So I have my T, my A, my G, and my A. And on my other strand I have A, T, C, T. So there's my two strands there. I'm going to grab a different color so you guys can see a little bit easier what is happening here. So what is happening is my nucleotides are coming in. I have my A coming in on this side. It's a bad pen. I'm going to throw that away. A on the inside here, my T on that side, AT, AT, GC, GC, and so on and so forth. Then eventually all of the DNA gets added along, so I'm going to rewrite here T, A, G, G, A. I'm separate a little bit more so you can see it a little bit better. A, T, C, C, A. Okay, and, oh, I made a mistake. T. All right, and what you're going to do on the inside here, we're going to add the complementary bases. A, T, G, C, A, T, T, A, A, T, G, C, G, C, A, T. So notice here, if I'm reading across like a book, they're identical. I have T, A, T, A, A, T, A, T, G, C, G, C, G, C, G, C, A, T, A, T. The code is exactly the same. Nothing is messed up there. So the last thing that we're missing, if you notice here, here's my backbones. Right, there's my backbones there. I have my two strands coming in. And notice that I don't have a backbone on the inside. Hi, Holden. Say hi really quick. Hi. All right. Step number 3 then is what you're going to do is you're going to covalently bond the new backbone along the inside. Okay, so you're going to covalently bond the new backbone along the inside. So what you're going to have here is just going to have your strands. Separate them a little bit further. I have my T, A, G, G, A, T, C, C, T. And I'm going to add my new ones here. And I have my A, T, C, C, T, T, A, G, G, A, and then I have my new backbone here. Okay, so at the end, you end up with 2 identical strands of DNA, 2 identical strands. Now we only really care about the coding. I know there are technically 4 strands of DNA here. We call both these one strand 2, but they're really, we'll call them copies rather than strands. So we have 2 identical copies of my DNA, 4 strands. You always have an old strand on the outside. Old strand on the outside. Those old strands are sometimes called the template strands. The older template strands because that's the template that you use. So you have your old template strands on the outside, and then your new strands are on the inside. So my new strands here are on the inside, and you can call those your complementary strands or whatnot. But it always goes old, new, new, old. I remember oh no, and that's called semi-conservative replication, because conservative means like to keep the same. So semi means half. So this DNA strand right here is half old, half new. This DNA strand is half old, half new. Now a common mistake many people make is they invert. They always add to the right, and so what you'd end up having here is one of your strands having this. I'm just going to view the bases. I'm not going to do the backbones. Remember, the code is the most important part. I'm just going to show you guys a common mistake that people make. Okay, remember the first strand is always the coding strand. So notice that this code here, TAGGA, is different than this code strand here, ATCCT. So this is incorrect. This is a common mistake. You always add your new bases along the inside. So there's really 3 steps to this, and I always tell these 3 steps to follow: unzip, add, and then glue. And so whenever you're doing replication, you always unzip and unwind first. You add your complementary nucleotides, your complementary bases, along the inside, and then you glue the backbones together, those phosphates and you sugars, you glue them back together. All right, that's it for now. Just 3 steps, 3 straightforward. You guys have a worksheet to work on that goes along with this. Sorry for all the interruptions in the background, but I'll see you soon. I think we have notes in another couple of days. All right, bye, Pirates. Hope you have a great day.